Lunch and Learn. It is Friday, January 18th. Today we have a special speaker, Dr. David Cantor. You've seen the announcements I've sent out. Just very quickly, David he's a brilliant clinician. It's a real privilege and honor to have him as a friend and colleague. He has done these talks before in various locations, including a webinar here a couple of years ago on neurotoxins and toxins in the environment. And he really knows his stuff. So I'm not going to give you much more of a long intro to say that David is also the president-elect of ISNR. And so David, thank you so much. I know you're a busy guy, and we really appreciate you taking the time to be with us today. It's all yours.
Yeah, well, thanks, Rob. Thanks, Rob and Richard. I appreciate the honor, really, the opportunity to share a little bit of information and everything. Yeah, as Rob had mentioned, I've done this talk a couple times before as part of ISNR, and of course this Lunch and Learn. And Rob had approached me a little while back and said that it's time for us to sort of rally around and bring it up again. And there's been a few, obviously, some updates and things, so that's what I've tried to include here. So let me go ahead and proceed. We're going to talk about neurotoxicity assessment and treatment concerns. In this context, clearly we know neurotoxicity can bring everybody down physically, but obviously as a mental health professional, we're looking at what these things do in terms of its effect on brain function and behavior. I have a couple of definitions, and I'm not going to go into the details of these things because we don't really even quite have an hour here, but the idea is to give you some general highlights and points, and hopefully we'll have a couple of minutes at the end where people can ask some specific questions. But neurotoxins are really any kind of quote-unquote nerve poison. It can belong to what they call an exogenous class, which means it comes from the outside of the body. And we'll talk about some of those. But recognize that our body is constantly producing various endogenous compounds, which are toxic. And there are mechanisms that our bodies use to get rid of those toxins as an outcome of metabolism. And that's what we do. And when anything is not working right, we can literally be poisoning ourselves from our own activity, which a lot of people don't realize. And so we'll talk about that. Neurotoxins really inhibit neural activity. They affect the ion concentrations across the cell membrane. They affect the synapses. They can actually cause cell death, and in a variety of ways, actually. They definitely impinge on glial cells. It's not just the nerve cells. There are the glial cells that are in the central nervous system, and as many of you know, glial cells play a very important role in the support process of neural activity, and we're learning more and more that it's more than just a support process. They actually play a critical role in the integrity of the central nervous system directly. So we do have some defenses in our bodies that help to protect us against toxins. One of them, many of you have heard, is the blood-brain barrier. This is made up of a protective set of cells, astrocytes that are around the capillaries as they come into the brain, and this facilitates things from our blood are absorbed into the brain, and if there is something that is potentially damaging, there is a blood-brain barrier that at least gives us some screening, if you will, from various insults. That's one barrier. It's not a great one. We'll talk about in a few minutes, but it is one of them. There is this other structure called the choroid plexus. Again, this is a vascular layer of tissues around the third and fourth and lateral ventricles in the brain. And they, again, provide a sort of little wall or barrier, which hopefully will be selective in what can pass through in the form of nutrients and other things, and trap other things like heavy metals not to get into the brain. So these are the classic, at least the ones that primarily that we have as some sort of barrier protection to hopefully not have things invade our brain. Most of the things we're talking about here are called hydrophobic, meaning they're water soluble. I'm sorry, and our lipophilic are fat soluble. And so when we talk about toxins, we're talking about certainly the more dangerous ones, which I'll explain, are the fat-loving lipophilic toxins. And because our brain is made up of a big fat sack of fat, really. This becomes a kind of a natural thing to get absorbed into brain tissue with fat-seeking toxins, and this is where the real danger is. Oh my goodness, this didn't come out real well when I transferred the, okay, so there are a variety of toxins. I said there are a variety of toxins here, and I just sort of list on the right some of the common toxins, but each of these different toxins has a different site of action. It can either affect stuff right at the cell membrane in the brain. It can affect the actual availability or metabolism of the cell, of the nerve cell. It can actually create and interfere with neurotransmitter production. So needless to say, there are many, many different types of actions that various types of neurotoxins can produce in a variety of ways to interfere with the efficiency of the brain functioning. Now, the other guardian that we have from toxins is the liver, and probably this is actually one of the most important. We talked about the blood-brain barrier. The thing about the blood-brain barrier and the choroid plexus is that these, and we'll come back to this, these fat-seeking toxins, they go right through those fat layers of those barriers, and so we don't really have much protection. The liver, on the other hand, is a structure, an organ in our body that converts various toxins from being fat-based or fat-seeking to become water-soluble. And once they become water-soluble, then they can go out through our urine and so on. So it plays a very important metabolic function in converting compounds in our body. It breaks down compounds. It is the primary site, for example, in which various psychotropic medications are metabolized. And so if you have liver dysfunction, lots of things become complicated, including, of course, our ability to properly utilize psychotropic medications. So it is another important structure that naturally helps to protect us. And there's a sort of detailed pathway that describes this, but I think I have a simpler slide here. There are also ways to measure the efficiency in which our liver is functioning to do this. And just so that you as clinicians or practitioners know that anytime you suspect that a patient may be not responding well to any kind of treatment, whether it's psychotropic or in this case neurofeedback or what have you, that there may be things operating in the background that are working against you or thwarting you, or maybe you get a certain amount of success but then the patient regresses. There are lab tests that can be done to measure the efficiency of what they call liver detoxification. This is just one example of that by Genova Laboratories. They have, as you can see, a detoxification test, which you can run, and it will tell you how efficiently the liver is doing its job with this process, and gives you a readout of where there's things that are not working well or they are working well, and then based on that, a proper health practitioner can begin to mediate the inefficiencies, improve liver functioning, obviously, and offset some of the buildup of toxins that may have occurred. This is the simpler picture that I wanted to show you related to this. So as you can see here on the left, you've got fat-soluble toxins. What the liver does is it uses various nutrients, and it lists those for you, some of the key ones. Anyway, you can see the important role of vitamins and folic acid, and one of the big names that some of you may recognize there is glutathione. Some of you may remember that years ago, particularly with autism, there was another new promising cure or improvement by using glutathione supplementation in autistics, because they found that glutathione was very low in a lot of autistics, and said, that's the problem. Well, not entirely. Yes, that's true, glutathione was low, but obviously there are many components and required nutrients, and one of the reasons that glutathione gets low is that as toxins build up and you're exhausting your availability of glutathione to help in this conjugation, it's kind of like a vicious cycle. Then of course the glutathione will register as low. But this also speaks to something we'll just touch on today, which is, as many of you know, is diet. And if you can't, if you're working with a kid in particular who doesn't eat well, doesn't get access to a lot of dietary important ingredients and what have you, you can begin to see that begins to weigh heavy on all kinds of things, but particularly in the efficiency of this detoxification process. And then it goes to a phase 2, and there are other requirements. And again, this just lists these things. So these are all amino acids and important metabolic cofactors that you really, only way you can get some of these things is through your diet. Your body cannot manufacture these things. So diet becomes critically important, as many of us know. And this is one of the reasons it does. And this is the relationship between when you are dietary, if you have dietary insufficiencies, you're compromising liver efficiency, which now can contribute to a buildup of various toxins, which then leads to brain dysfunction and all the things that go with that. So this is a set of pathways. You can see that the brain does not operate independent of the body. And this is one reason that as practitioners, where we focus on behavior in the brain and neurofeedback, we really have to be at least aware as whole body practitioners. We have to kind of be aware of these other things. So as I said, this conjugates the toxins. They go out as now water-soluble waste products, which now can go out through various other organs or mechanisms. And this is the way we get rid of toxins as one of the key defense processes. So this is an example of, for every molecule of pesticide we get exposed to, and we're all of are getting exposed to pesticides in one way or the other, we're actually depleting a molecule of glutathione, magnesium, and other nutrients. So we're constantly burning up our storage, if you will, of these key nutrients to maintain liver functioning and efficiency to protect us. And if we don't supplement those nutrients, we're going to continue to create, in the course of a lifetime, compromise. This is a slide illustrating just how potent this is, particularly as it relates to autism. This is from a couple of publications many years ago that I did with Steve Edelson, where we did a blood analysis of the levels in children, and we actually did 2 independent publications of almost about 30 kids, 30 autistic kids each, and they replicated very well. And this is not all of, you see on the x-axis, the various, these are lipophilic, fat-seeking toxin compounds. Many of them are in common products like the glues that keep our furniture together, or products that are in the furnishings of our home, our carpet, etc. So we get exposed to these things constantly as airborne particulates and things like that. Well, in the blood of these kids, what these numbers mean is that you're seeing some measures like, I think the lowest on here is about 183 for gamma chloridane. 183 means it's 183 times the MAP as a percentage, 183% above the adult maximum level of tolerance for that compound. So all the way up to chloroform, which is in this case, and these are averages now across the number of cases in this initial study, 2,000 times the adult maximum percentage level of tolerance for that. So these kids are walking around with extraordinarily high levels of lipophilic toxins floating around in their system, so to speak, constantly wreaking havoc on brain function, brain development, and other things, of course, in the body. This is another graph from another study done also by Edelson a number of years ago, looking at the levels in parts per billion and solvents related to ALS. That's Lou Gehrig disease, alateral sclerosis. And you can see the comparison of the patients with ALS versus the black bars represent what should be the control group here. In virtually, well, not virtually, in every single case of the various, and there's 10 different solvents being listed here, and they're listed there for you, these ALS patients are way significantly higher in what their body burden levels are compared to the control group. So it's another illustration of the potential role of neurotoxins in a lot of these neurofunctional and neuromuscular, neuropsychiatric disorders that we talk about. And this is why we think it's an important issue for everyone to be aware of. So these things also play a role with the immune system. And we always ask, why is the immune system seemingly to be relatively ignored by the mental health community or mental health, which is always a mystery to me, because in the American Psychiatric publishing of the textbook on psychopharmacology by Schatzberg and Nemiroff, you'll notice that chapter 8 is an entire chapter dedicated to the interaction of the brain and the immune system and its relevance to the treatment of neuropsychiatric disorders. I mean, if this is in the textbook of psychopharmacology, so anyone that deals in the world of treating mental health disorders with psychopharmacology certainly should be sensitive or aware of the brain immune system connection, and yet we hear relatively very little about it, certainly in private practices, where I certainly don't run into too many psychiatrists who even think about those things. But here's the relevance of this. The immune system kicks out these proteins called cytokines. Many of these cytokines are pro-inflammatory. They aggravate, and again, they can be further triggered by elevations of toxins in your system. And so there's a relationship between the toxins, your immune system, and then what all of these things do to brain function and brain development. This is a little flow chart to give you a sort of quick idea that we get exposed to these things either, even in some cases during gestation. We can talk about that. There's quite a few studies to show that what the gestating infant gets from the mother is already wreaking havoc on aspects of brain development. So right from the get-go, so to speak, as a young infant, there are already aberrant processes occurring which will lead to developmental problems of one sort or the other. But all these things directly or indirectly can affect the brain, as you see, and ultimately it can lead to a variety of types of dysfunctional issues that can lead to what we call now autism spectrum disorder. So in the last 10 years, there's been a lot of evidence to show that these pro-inflammatory cytokines do exist in the autistic populations. There have been lots of researchers to show that these things cause inflammation. So we do that. The general mechanism as a result of being exposed to these things causes inflammation in the brain. And primarily it attacks the glial cells. So as these things become dysfunctional, depending on what areas of the brain are affected, we can get a variety of problems that will result. And as pointed out in this paragraph, a lot of this can already happen because the mother has some sort of immunogenetic factors that, again, contribute to this process. So again, this is just another slide talking about the fact there is work done going all the way back to the 1950s by Roder, who looked at when mothers were taking thalidomide, and that drug actually alters the genes in a way that most of those children, it was argued that these two children were born with, limbs missing or some sort of body dysmorphia. The actual fact is, a few people know all of these children had autism. And it's another kind of piece of information that gives you this idea that once you're exposed to something that's toxic, besides all the other things that I've mentioned, it's actually altering our DNA. And this is why now you have a genetic carryover. I often tell the little story about, I worked with some toxicologists who are working in Lake Erie, which, as many of you know, has been always been a festering problem with toxins that have built up in the Great Lakes. And he was studying fish that were exposed to the toxins in Lake Erie. And he pointed out to me one day, he says, if I take this fish, this one fish who's been toxically exposed out of Lake Erie, and now I put him in a nice, really clean lake, he says, despite the fact that this fish and all the offspring for the next 7 generations will no longer be exposed to toxins, the effect of this initial toxin exposure from Lake Erie will still be felt 7 generations deep because of the restructuring or impact that it does on the DNA of that original fish. So the issues are not just, in a sense, unidimensional for a single generation. This is where this issue is huge, because what we're doing to ourselves, frankly, have been doing to ourselves for many, many decades, is already probably why when you study the epidemiology of disorders like multiple sclerosis, like autism, like we can go on and on, it's not just what the current or immediate exposure within one generation, one lifespan. We're actually seeing potentially that more and more people born have already genetic predispositions or susceptibilities, we'll call it, to lead to these immune genotype alterations that make us more susceptible to have any number of neurofunctional and neurodevelopmental problems. So again, this cytokine can also be promoted by gut factors, where the bacteria in the gut is influencing this. And this was already recognized by the APA in 2012, which I was shocked by when I saw this on the front cover of the Monitor in 2012, where even the APA, which I've considered not always in touch with these things, but they are. And this was a focus on the gut. What happens in the mind-belly bacteria connection that, again, can contribute to our behaviors and the things that we examine. Well, GI inflammation, or what they call gut dysbiosis, affects this process. And again, cytokines are enrolled here. And of course, this blocks or prevents the ability to get access to neurotransmitters. They've done lots of animal studies to show this with a change of social behaviors in rats. Again, when you mess around and you affect the bacteria that our bodies really depend on, which reside largely in our gut, there's huge ramifications for how it potentially affects the brain and behavior. One of the things as a developmental neuropsychologist, I frequently ask all my patients and parents, of course, is their exposure to generalized antibiotics. As you know, general antibiotics don't just kill the bacteria in an ear infection or a sinus infection. It destroys the bacteria that we have in our gut, for the good bacteria that we need for so many different reasons. One of those purposes of that gut bacteria is to fully decompose foods that we get exposed to. And if that becomes compromised, when these children are taking 3 and 4 rounds, oftentimes in the course of a year, of a generalized antibiotic, the gut really never has a chance to fully recover or repair from those insults, we'll call it, and they can develop a condition called leaky gut syndrome. And what that means essentially is the gut now has been compromised, so many times food particles are not fully decomposed. They get into the blood. The immune system comes along, thinks it's an invading infection, and now develops an antigen to a food, and this is how we develop food allergies. Well, when a child, and young children, of course, the common things they eat are crackers and cookies and certain wheat products, and of course a lot of dairy products. So now you get a lot of dairy and food allergies promoted by this process, and it's all because of our overextensive use of generalized antibiotics. So this is another concern when we look at some of these things. It affects our immune system, our autoimmune system. This is also a little fact that is relevant, and even in healthy, or generally in healthy individuals, about 95% of the peripheral 5-HT that we have in our bodies, which is a primary protein set that we use to produce serotonin, for example, resides in the GI tract. So people have argued that there's a possible link between the GI tract inflammation resulting in or being caused by an increased local 5-HT compelling based on the connection between serotonin, immune system, and nervous system. In other words, if you are unable to utilize factors that are needed to develop and utilize the the development of serotonin, as we get serotonin deficiencies, we become what? Susceptible to all kinds of mood regulation issues, sleep problems, etc. So this is, again, a factor that may be contributing to that. The Russians, I have had a phase in my life where I've, had to, and wanted to, of course, read quite a bit of the Russian literature, which was not available to us, certainly not during the Soviet regime. But they have done a lot of studies to look at the impact of these factors, like cytokines, on neural firing rates. And so the relevance of this, as people who look at EEGs, obviously, and what we do, when we're looking at things like excess theta or excess delta or slowing or what have you, or some aberration, we'll call it, of frequency firing of neurons, recognize that everything I'm highlighting for you today can alter the neural firing rates in various structures, part of networks in the brain, depending on, again, what those regions of interest are and so on. But when we see abnormal firing rates, which is what we're treating with neurofeedback a lot of times, we're trying to alter and balance that back out, recognize in some cases, this is potentially, the things I'm talking about is how it got there, and recognize too that while you're trying to correct or compensate for these abnormal firing rates, is what we do in neurofeedback, as long as there's a pervasive or ongoing chronic condition which is promoting the changing the firing rates, we're working an uphill battle here. And again, this is where my message to you today is being aware of these things, that when we don't get as successful or as fast, or in some cases any complete good outcome with what we're trying to do, and we do compensatory therapies like neurofeedback, these are factors. This is a good reason why I've been arguing for a while now why we as mental health practitioners need to buddy up with our functional medicine docs. There's a lot of things that we can share and work together to reach a common goal and use the tools and knowledge that we have respectively. This is an example of a study published a couple of years ago that I did with Dan Tuttle and Richard Lord in a case study to show what can happen when the bacteria do get imbalanced. Now, again, I won't go into the details of this, and this I do have in the slide set if somebody wants to look at some of this, the details of how we measure these things. But what we were looking at here is this is a set of symptoms that this case had for all of her life. I mean, she was around 70 years old where we finally got into this, and noticed a lot of these physical symptoms, I would call you, but I will tell you that there are some comorbid mental health things that kind of go along with this, and this is not unusual when you physically don't feel well, you have some low tolerances for stress and other things. The number is sort of a rating here of how often or how severe these symptoms were occurring, and the post is after where we did a what's called a bile salts substitution. In other words, what we found here was that this woman had thank you. This woman had a genetic defect in her body's ability to produce a bile salt, which helps keep the balance of bacteria in the gut going. And this comes from, I think it was from the gallbladder. And so the net effect is, let me show you here. This is the distribution of the various key bacteria before BART, which is the bile salt replacement therapy, and afterward. And you can see huge changes in the proportion of certain bacteria in the gut as a result of properly balancing with a bile salt. Okay, and we tracked this after 3 weeks. We tracked it after a year. So the importance of this now can be illustrated in our brain maps. The QEEG brain maps that you see on top here, the lighter the color, so to speak, the more abnormal the deviation. And naturally, what jumps out at you here is notice on the top we have excessive theta, which is fairly diffuse. Some areas obviously worse than others. But we're talking, for the most part, most of these measures are clearly over 2 z-scores. There are other elevated features in some of the other frequency bands here, but this is an example of that excess slowing that I was talking to you about, and there are concomitant effects, of course, with behavior and functioning. 3 weeks after the supplementation, look at, on the bottom set of maps, a remediation of those excesses, where virtually all, or certainly most of the measures now are well below 2 standard deviations of 2 z-scores. And all that took place here was a correction in the bioavailability of a bile salt to get the bacteria balance corrected in the gut, which had concomitant effects. That's the only thing that happened here. And after 70 years of having symptoms, in 3 weeks, her symptoms start to lift, and it's illustrated by changes in brain function. And we see the same thing in our Loretta maps. Again, on top, you're looking at the z-score Loretta maps showing the deviations in a variety of ROIs. Most of this is in the theta band on top, where we're seeing excess theta, and then we're seeing deficiencies in beta. You can see the actual narrow bands here listed at 7.35 and 23.7 respectively. So this is a theta-beta imbalance. Some of that should ring a bell with you. And you can see how diffuse it is. Now look at the bottom set of Loretta maps, and you can see huge normalization improvement after the BART intervention. So this was one case study, of course. It's not going to convince anybody about anything largely, but it gives an illustration potentially not only of the role of what happens when, for genetic reasons or otherwise, we get an imbalance in bacterial distribution in the gut, and how it can affect a lifelong set of problems physically and mentally. But where it also, we can measure it now with the tools that we've had in recent times. We can see the impact of this issue on brain function. If someone came to me and said, and I looked at these brain maps and said, oh yeah, this has got excess theta, we know what we want to do. We want to treat this with, trying to inhibit theta in certain regions, or elevate or improve or reward beta, and so on, getting a balancing process going without even knowledge or regard necessarily to how it got here or why it's here. But I can almost predict that this might have been a bit of an uphill battle in this particular case, because we have an underlying physiological process that is, in a sense, chronically driving this condition to be there. And by being aware or looking into, or at least considering other factors that may be going on here, particularly when we look at some of the symptom lists that I talked about or showed to you earlier, again, we, through a functional medicine set of tests, identified this, came up with an intervention that was fairly straightforward and easy, and you can see the effect. One of the other things of more recent times that I've been noticing, and of course, I'm sure you have too, is we see it on the 6 o'clock news about foodborne diseases. These are diseases where we're hearing about people getting very ill because of the foods they're eating, because there are, again, bad bacteria infections, if you will, and in some cases it's been fatal. So some statistics here that we're talking about 76 million cases resulting in 323,000 hospitalizations and 5,000 deaths each year is due to foodborne diseases. 1 in 4 Americans, 25% of the population of this country, will likely develop a foodborne illness at some point. And out of that, 1 in 1,000 of Americans will be hospitalized as a result of this process. So it's a huge cost factor. This all came from statistics from the CDC. This shows you some of the more common type of foodborne illnesses or bacteria, and I've broken these out for you in terms of which ones are the biggest culprits. Salmonella, of course, is the biggest, and we hear about that most often in newscasts and broadcasts, what have you. Toxoplasmosis, if some of you may be aware that that is a major factor, and for those that study toxoplasmosis, there are variants of that that we know for a fact alter, in a sense, capture control over areas of the brain functioning, driving things like risk behaviors and unusual anxieties and all kinds of things like that. So again, the whole process, not only where we talk about toxins when we think about metallotoxins and these pesticides, our own foods themselves carry bacterial content potentially that have ramifications on bacteria infection and bacteria alterations. This chart, again, from Mead et al., published in 1999, is showing you the number of cases at that time where these kinds of bacteria are already coexisting, co-hosted, if you will, by either humans or other kinds of animals, and the percentage as a result in food. Recognize that if it's in the animal, in many cases we'll get into this because we eat pigs and cows and these kind of animals anyway, poultry. So again, there is a food chain effect, contamination issue, which a lot of people are not aware of, and this is potentially how these agents get into our bodies and ultimately affect us. I've alluded to the fact that some of these things are already happening prenatally or perinatally, and this is when, of course, humans are most vulnerable for a lot of these things. There are some studies done by Swedish researchers who have now published several studies, again, looking in the amniotic fluid, the presence of inflammatory cytokines, that these are markers for already setting up immunological dysfunction in children who develop or exhibit autistic spectrum behaviors. This is just another case in point reference, if you will, of where they're identifying these things prenatally in the mothers. In my work with a former top Soviet at the time, I was doing some work while the Soviet Union was actually the Soviet Union, with a top pediatric toxicologist out of Moscow in the Soviet Union, and he was collecting with all of his clinics and all around the Soviet Union, getting information about various excess elements or excess concentrations of various toxins during pregnancy, labor, looking at the placental material and so on. Huge amounts they were finding in the Russian population, I should say Soviet population, that already were certainly predicting or certainly could be high risk factors for what happens with the children. We've even found that maternal thyroid hormones are essential. We know that thyroid plays an effect on brain function. We see it all the time, and it affects certain EEG characteristics and again can be correlated to certain behaviors. But at the same time, it also again can have a disruptive effect on development in the children. So anytime, again, as a pediatric neuropsychologist, when I'm getting information, I'm very curious about what's the thyroid history in the family. Does the mother have a history of thyroid problems or other kinds of, again, genetic predisposition, thyroid dysregulation issues that I need to be aware of? I'm always looking at the things that might hamper or thwart or give me some unusual kind of response to what I would regard that should be a pretty successful protocol in neurofeedback. I'm looking at what might get in my way, and these are some of the things we're talking about. Again, pesticides and these lipophilic compounds affect hormones. They affect thyroid. And so, again, these things are interactive. When you start talking about the toxins, they're basically hormone disruptors. And there's a huge, huge body of literature that has been emergent over several years now, looking at this more and more carefully because neurohormones and other types of hormones that play, again, a huge role in mood and brain development. So the moral of the story is, with your clients, get good history. It's just really, really important, not only to help with getting a more successful or potentially more successful outcome in terms of your success working with your clients and using neurofeedback and other forms of neurotherapy, but also to prevent a recidivism, a regression. You get them there and they come back a year later and say, golly, we're getting our symptoms back again. And so we don't get the hoped for permanent solution that we certainly know about that can happen from neurofeedback because these things are not being addressed or looking at. So what's depleting the immune system? Again, air pollution, pollutants that I said, particulates in the air. Again, the APA was on this with one of their lead articles. Again, I don't remember which year this was, but again, there's a clear recognition that there is a poisoning of the planet, if you will. This is some old data from many years ago. Again, this is increased, not just affecting with physical health like cancer and the things we hear about. I was in a huge meeting worldwide in 1994 of delegates, US health delegates from all over the world that met in the former Soviet Union at that point, because the Soviets came down, or ex-Soviets wanted to invite delegates from all over the world on what they have actually been doing for half a century in terms of their mismanagement of, well, like the Chernobyl accident. It wasn't one Chernobyl accident, it were 5 Chernobyl-like accidents. There wasn't just, this accident. There was huge. Remember that the Soviet Union at one point had control over 1/8 of the total land mass of the planet. And so with that meeting was to sort of, bear up and say, let us really tell you what we've been doing, and we're now asking for your help in trying to figure out what's the risk issues now that's going forward into the 21st century. I will just summarize and quickly say that it's too late. The horse is out of the barn, out of the field, over the next hill and into the next town. There's no way to get the horse back. The damage has been done. And so a lot of the epidemiological factors we see worldwide with a lot of health issues in general, and I argued as the only neuropsychologist at that conference that, for whatever physical health problems that these things contribute to, I would say that there's probably 1,000 times more people that would be affected on the mental health issues just because of the subtleties that these things create in the neurochemistry of brain functioning and brain development. So again, this is something that has been recognized a long, long time, actually going all the way back to Youthon, who was the General Secretary of the United Nations, who wrote a book on this back in the late 60s. And again, it went largely on deaf ears. Again, there have been several studies to look at the exposure issues of women here that you see from Wu et al. To air particulates and how it affects the cognitive decline. The correlations of these things are very high. Animal studies, of course, have been done to look at the same when you get exposed to particulates that are in the air from smog and fallout in certainly major city areas. What does that mean? Not too long ago, there was a major fire here in Atlanta that was all over the national news that destroyed a bridging structure on one of the major intersections around Metro Atlanta. And of course, you created traffic problems to an already horrific traffic management situation in Atlanta. A lot of people didn't realize that the PCP burning, I was more concerned about the fumes and the fallout from heavy smoke. Thousands of people are sitting trapped up on this highway, breathing in all these particulates. Nobody seemed to be concerned about what that risk factors were going to be to these people over the next several years, having been exposed to that. They were more concerned about how it was going to create traffic problems for people. There's a large correlation of ADD and ADHD to allergies. Again, this is just more research and stuff that shows that when the immune system has got sensitivities or prone to having sensitivities where it may kick out these neuroinflammatory cytokines and other things that will affect brain function. This is some studies that I did with Doris Rapp and some of her clients. Doris Rapp was a pediatrician allergist that published several books and was on TV a lot in the 1980s and 1990s. But we actually did placebo-controlled studies on case studies where we looked at various factors that were affected with the immune complements before and after being exposed to an allergen, and a huge change, and ultimately it affected brain function. Another study of what's called 2378 TCDD in Times Beach. This was fallout from dioxins that was spread out throughout the town outside of in Times Beach, Missouri. And this is just a quick couple of slides to show you that once you expose to these toxins, you get a damage done to the immunoglobulins. You can see the difference between this is for children that were exposed, by the way, gestationally. So we looked at them 15, 16 years later, and this is what it ended up doing to this group. And then we look at the number of neurometric or QEG abnormal hits distributed. The largest proportion of abnormalities in terms of the percentage of hits is in the frontal lobe. Why? Because as we all know, the frontal lobe is slower to develop. They're more vulnerable to these intrusions happening in early life, and this is the end result of that process. There are many studies looking at the relationship between ADHD and urinary metabolites of organophosphate pesticides. Again, I'm not going to go through all of these. I've done a lot of work and also shown on the effects of toxic lead exposures and have testified in court. And in fact, in 2012, testified as part of a multi-case study there of the largest legal case in the history of the state of Missouri, in which we demonstrated that early exposure to lead in children did long-term damage to brain function and the lives of so many people exposed to lead from this lead plant. Because of this information from the QEG stuff and other things, the defendants were awarded $500 million between compensatory and punitive damages as a result of that demonstration. There are lots of sources. Again, this will be in the slides or other sources if you want that I can send to you for where these toxins. We think about that a large portion of the lead has been taken out since we used unleaded gasoline, and that actually was one of the impetuses for getting lead out, was to have a lot of testimony by people to show evidence that this had potential damaging effects, particularly to children. But now still, in a lot of our kitchenware and other products that we have probably around our home, these products contain trace amounts of lead that get exposed to it. Again, in our clinic, I got rid of all the toys that we used to leave out for children to play because they're produced in China. The dye that's used to do the color in a lot of the toys actually contains lead, and I had a group come in and actually measure the lead and trace amounts that I was actually inadvertently, obviously, when the children play and put it to their mouths and what have you, they're getting exposed to small amounts of lead. And I said, well, that takes care of that. We're getting rid of this. So again, there are biomarkers of ways that that these issues can lead to. We already know drug exposures, prenatal issues become where we have cocaine-taking mothers. They affect the dopaminergic system. Leslie Pritchett, one of my partners, has done extensive work through the National Institute of Drug Abuse looking at the effects of these things with QEG measures. And again, these are another sources of, in a sense, a toxin-related issue that affects brain chemistry. Can we clean up the environment? One of my takeaway messages here is, I kind of, we know that there are water treatment plants around certainly major metropolitan areas, and maybe like you, I get these annual reports of what my water system is in my home and what they're doing and how much is left from various fluorides and other agents. And none of these treatment plants are 100%, as you know. And by the guidelines or the cutoffs of the EPA, they're saying, and you're drinking healthy water. I can tell you assuredly, not only you're not drinking healthy water, and there are plenty of studies for some of the compounds, even for the small residual amounts that are left here, that can build up in a system because leaving it with what the treatment plant does for your public water is not the way to leave it. And that's one of the arguments, that actually the agent that's used to treat the water is a problem. Trihalomethane, which is a substance used as part of the treatment process, is actually a toxin. And this is one of the things is that again, there have been a few studies that have come out on this, and here's a study that shows that the impact of chlorine or chlorinated products, which they use to kill these so-called bad bacteria in the water to protect us from, again, bacteria infiltrated water. The products they're using are toxins themselves, and then we get exposed to that. So you're trading off trying to protect the population of one thing, but the very agents you're using to control are toxins in and of themselves, which again raise the risk of the neurodevelopmental disorders and other kinds of problems. So in our mad scramble to supposedly drink cleaner water, it doesn't look like it, bottom line, and we have these ongoing risks. 2 in 5 children in America show signs of fluoride poisoning. That is an issue. The agency from the U.S. Centers for Disease Control concluded that fluoride levels, fluoride is in a lot of water systems, New York being one of the major ones, of course, and been doing this for decades. And the assumption was, well, it's not fluorine. That fluorine we know is a toxic. Fluoride, which is in our toothpaste and obviously used in various water sources. But I'm telling you that this is a toxin. not on the same level as the natural element fluorine, but because it's a derivative, but it definitely shows data showing that this is an issue. And as we lead into the 21st century, we're finding that our own uses of EEG and quantitative EEG uses actually can be used to measure these things. We actually can see these things as an outgrowth of exposure to some of these agents that we're talking about. And this is a little table that does show the impact on theta amplitude, for example, alpha amplitude, and so on, gamma, all the way through gamma, for the various products and what the relationship ultimately is with the exposure or the body burden level of these exposures can lead to the effect on the EEG itself. So again, we actually are already using tools that are potentially sensitive to recognize when there may be a potential contaminant or effect from any of these sources I've talked to you about today. And all I'm sharing with you is, since we do this naturally when we do neurofeedback and our baseline assessments and so on, it's good to try to maybe just keep in the back of your mind that because of the patient's history, the family history, medical history, exposure issues or potential. I had a lot of adopted children that have been adopted from Russia that come to me with various neurodevelopmental issues with ADHD and what have you. And knowing what I know about the geographic distributions of some of the things, I had an opportunity to see that in the 1990s, it doesn't surprise me. Now, a lot of the children that are adopted out of some of these geographic regions are going to continue to have issues or concerns of developmental problems. So one of the last takeaway messages here before I give you a couple of minutes to ask questions is I'm suggesting there are a variety of what I would call functional medicine tools, or at least domains to be considered. When we look at the whole issue of the brain function related to behavior kinds of issues that we do as therapists, and recognize that we do have ways to look at toxins. We clearly have ways to look at things that we call oxidative stress profiles. I know Richard has been looking at a lot of that for a long time and has presented some great stuff on that. But recognize that the sources of these problems potentially come from these things that I've shared with you today. And hopefully we'll all be more aware of these things and at least take them in consideration as we continue to see patients. And hopefully as we go forward as a body of concerned health professionals, that we become more both organic and integrative with our fellow practitioners, like I said. And I know one of the missions we're looking at ISNR is trying to build more bridges with the functional medicine docs and experts out there and the biochemists, because a lot of what they do, what they assess, and the treatments they offer can very nicely be integrated and should be at least considered with a lot of the work that we do when we're trying to compensate brain abnormalities as it relates to behaviors. So that's my overview. I apologize for kind of blitzing through that, but I think we have just a couple of minutes here if anyone has any questions or comments.
Well, thank you, David. Let's see if some folks bring up some questions. I know that Richard's unmuting them.
Dr. Cantor, this is Martin. I have a question. How does this information flow into the realm of public health authorities? That's where it should be in my eyes, since we need to get at causes of all this.
I agree with you, Martin, and let me quickly share. Yes, you're absolutely right, but I also want to let you know, at least in this country, it's very much a political/economic concern. For example, there is a big plant, and here in the South where I live, one of the major industries is wood processing. Wood processing produces a lot of metallotoxics like arsenic. I've actually run studies and have presented this. So you present this and go to the local public health people and say, look, we really have to do something about that. And of course now at the higher levels, they say, what do you want us to do? Shut down a plant that employs 2,000 people in a poor rural area where suddenly people have no way to make a living? And suddenly it becomes like a political economic issue, recognizing that there is another issue that's a health issue. But there's a political economic issue where they say, yeah, but how do we make a transition where this is playing a major role in the people's ability to make a living, to live? So all I'm sharing with you is that I think on some level, people we'll call it the political and governmental structures, are battling with how to make a transition to get away from this without creating huge ramifications on a political economic scale that also will be detrimental for a lot of families in certain geographic regions. I mean, that's the best way I can answer that. Good question, though.
It's the same up here in Canada, where wood processing, of course, is a major industry.
Yeah.
And we have the same in the pharmacological industry, the same problem.
Yep.
And in the electronic industry with wireless and stuff.
Yep. So the reason why these things seem to persist decade after decade after decade, it's not because people aren't aware or at least somewhat sensitive. It's we've got ourselves so entrenched in these industries and the roles that these industries play in our daily lives, politically and economically, that making a transition can't happen very quickly. I will say I'm encouraged that we keep seeing more of the quote unquote green movement, particularly in the last 15 or 20 years. But as somebody that's been looking at all this for almost 45 years, it's long in coming, and it's still not, obviously it's not there yet. It's a very slow process to make that transition.
Thank you.
Yep.
Yeah, I have a question, Dr. Cantor. My name's Diane DiGiovanni. I'm seeing more and more people that are coming in to my office with anxiety, and also hearing about friends, their children are anxious, and young adults, they're anxious, and college students are anxious. So if you had somebody who had a chronic anxiety condition, what would you say would need to be tested first? I mean, I'm looking at the molecular level evaluations. Do you go into your doctor and say, first, try to find a functional person, of course, but do you go in and say, I want all of these, or do you have any correlates with certain conditions, with certain tests that would be helpful?
Well, look, practically speaking, if we had the ability to enable people to pay for all the tests, you'd say, let me make sure I'm not missing anything, and you run a broad-based spectrum of tests to be done. But practically speaking, that's not what typically is done, nor can people afford it, because much of this is not always paid by insurance yet. So what happens is that most functional medicine docs, for example, when they come in and they talk about their anxiety with you and what have you, when you refer them over to a functional medicine doc, they're going to go through their checklist of symptoms and history. And as some of them will call it, it's peeling back the onion. And, we begin to look at the longevity, the severity of certain symptoms, again, factors related to their diet, to their style of life, to where they live, to exposures. I mean, it goes on and on. And usually what the doctor says, let's start with some, sort of from the 30,000-foot view. For example, there are porphyrin tests that you can run that says there's clearly a marker for inflammation here or some sort of toxin thing. You don't know what it is yet, but if that shows up positive, that argues why I need to dig to the next level and start, peeling back the onion to find out more specifically what toxins are we talking about, what mechanism their inflammation. As you get more and more information, they are then able to say, okay, what we have to do is this to detox, we have to do this to fight off the inflammation, in healthier ways, et cetera, et cetera. So I'm saying it's part of a process. I don't profess to be an expert in that. That's why I obviously interact with other, with functional medicine docs. But this is their way of approaching it. Not unlike as a neuropsychologist, when somebody comes in with a certain set of symptoms and what have you, I've got to peel back. What are the functional components that lead to a reading problem, or lead to anxiety, or lead to what have you. And there's lots of different brain processes that can lead to any of those problems, and I need to find evidence that supports that hypothesis. So it's a process. I know that's not a simple, quick answer, but this is why I always tell people, stop looking for the quick answer. If you really want to do things, you have to do them methodically. And this is, as I said, this is what good functional medicine docs do as well. They'll take it, step by step, and peel back the onion to hopefully eventually find the mechanism and where the real source of the problem is, and fix it at its source, so to speak.
Okay, great. Thank you.
Okay.
David, one thing that you bring up, finding a functional medicine doctor, is the biggest complaint I have with neurofeedback practitioners. As you know, like you, and because I learned this from you, we promote, that people go out and try to find a functional medicine doctor when they're seeing signs like on the physiological checklist we have, and in the brain map, the patterns that we see for liver function, dysbiosis, and thyroid issues. Those are clearly on the maps. And so, the people that I train, the people I talk to, hundreds of clinics, say, we can't find anybody who does this kind of functional work. Where do I find them?
Well, it's the same, Richard. It's the same thing as, when somebody says, I'm trying to find a good neurofeedback therapist. Where do I find them? Okay. I mean, there's lots of people out there, obviously, that do it. And these days, of course, we tend to say, well, I don't know your geographic region, but a place to start with is to contact ISNR and other ADP, PB, and places where there are certified or well-trained or certainly recognized practitioners that can be listed. In the same capacity, we look at there's the Institute of Functional Medicine, the IFM, which is one of a couple of bodies that train and certify health practitioners and physicians in particular in this field. And so, one of the things I try to look at is, am I going to a physician that can show me that they've been through that certification or that training, or, in other words, like anybody else, do a little bit of due diligence to at least get a sense that this person probably has been through some recognized process of education and training that I can feel comfortable that they know what they're doing and that they're guiding me the best way. And I guess that's the best way I can answer that. So
I would agree with you, David. If you go to that website, the Institute for Functional Medicine, and you type in your state or area, it may not be close by. You may be driving an hour or an hour and a half, but just searching around in North Carolina, there were several people.
Yeah, the good thing I will say about functional medicine as a practice is, beyond maybe the first baseline assessment and some initial labs, a lot of the ongoing follow-up from that and the treatment, it doesn't require you to, like they do with us, of course, where you're doing weekly visits to somebody. A lot of this can be mediated with once-a-month consultation, sometimes just doing phone calls or now telehealth issues. So that the whole burden of having somebody really local to do it is not necessarily there in functional medicine as much as it is for other kinds of practice. So,
great answers. That really helpful. Thank you. And I know everybody's going to appreciate that help and how they need to do their due diligence. Well, we're at the end of the hour, so I guess we got to get everybody back to work. That was just stellar, David. It's, every time you do it, it gets better and more disturbing.
Sorry.
But it really shows you how much we have to look at the confounds, to neurofeedback, and it makes us feel good at, with all the clinics we work with, that we promote this point of view, and you really provide the justification for urging other clinicians, to look at the physiological roadblocks to a successful neurofeedback outcome. So this is just terrific stuff.
Well, thank you, David. Thank you for having me. It's always a pleasure, and you always have such a great group here. So I really enjoy it. So
if you're willing to share your slides, shoot them to me when you get a moment, David, and we'll get them out to everybody.
Great, no problem. Thank you.
Okay.